Oh, wait, and I also realized I haven't gone to the full presentation mode. Here we go. There we go. And I can switch back to text too. Okay, sweet. Okay, so I was going to do a brainstorm with you guys about the scientific process, and I'll still let you guys do that in class. Obviously, I won't be able to hear you, but I wanted to ask if you guys know what are the steps of the scientific process. And I'm not talking about specifically developing a vaccine. I just mean from point A to point Z, what are you guys thinking are the steps that we need to do? Absolutely. I'm so glad that they said ask a question because, and do research. Those are my favorite suggestions. And I think that is what I will emphasize here. So I made this little flow chart to illustrate the steps of the scientific process. And to begin with, I'm so glad somebody said ask a question because that's the most important thing. You see something you don't understand and you want to know more about it. And that's exactly why I went into studying microbiology, because I wanted to know what was going on in the world around me. So it all starts with asking a question. And then you got the second one from there. Doing research comes after that. And then what we have next is constructing a hypothesis. However, I do want to point out that a hypothesis can be really problematic because as the flawed creatures that we are, humans have a tendency to search for, interpret, and recall information in a way that confirms or supports our prior beliefs, which is known as confirmation bias. We have to remember that as scientists, our objective is to search for the truth rather than getting distracted by affirming our own ego. So it's okay to be wrong. If you're wrong, that just means you have to go back to the start of the process and you're gonna find more answers and you're gonna generate a lot more questions, which is the goal. We should be getting closer to the truth rather than just trying to tell who's right and who's wrong. So from there, we're going to see that you're going to construct an experiment to test your hypothesis, and you're going to figure out if that experiment produces data that aligns with your hypothesis or not, and you may have to change some things and go back to the start. But I also want to emphasize the last step of communicating the results, which is also very important in the process because the scientific process relies on peer review to discover whether or not somebody's making something up or this is the future of science. And without communicating those results, then we're never going to know what's true or what's fake. Okay. A great example of curiosity-driven research that turned into the most successful vaccination campaign in human history began in China in approximately 1000 AD, before the concept of microorganisms was even understood. Despite that, physicians did have a very good understanding of the spread of disease and found that if the scabs of an individual infected with smallpox were ground up and blown up the nose of an uninfected individual, it would protect them from future infection. I should say that this process, known as variolation, was very dangerous compared to vaccination. Although it did give somebody a chance for their immune system to build up tolerance to the disease, it also ran the risk of them having a full-blown infection and potentially die from it. However, later, in 1796, Edward Jenner met a milkmaid named Sarah Nelms who had contracted cowpox and had fresh lesions on her hands and arms. He developed a vaccine using matter from her lesions to inoculate a local boy named James Phipps with the milder cowpox virus. Jenner succeeded in making a vaccine which protected the boy from smallpox infection and would go on to reach the entire globe, allowing the World Health Organization to declare smallpox eradicated in 1980. Ironically, it is the eradication of infectious diseases which makes people skeptical about their necessity to this day, which leads me into my next topic about vaccine hesitancy. So some key events which have caused public distrust of vaccines have been the 1997 paper written by Andy Wakefield, in which he incorrectly correlated vaccines with the acquisition of autism, which was quickly retracted due to a gross misinterpretation of the data and ethical conflicts. So to this day, there is absolutely no evidence that vaccines, can I go back? Can you, yes, wait, I'm gonna need some more from you, Rebecca. Use their names just to keep everyone focused. I'm gonna randomly call out your names and I'm gonna ask you questions, I guess. So I know your names and I will call you out. Ewan, Patrick, Chris, I see you. I'm just joking. But let me go back. So on the topic of this paper by Andy Wakefield, he did correlate vaccines with autism, and this was a huge scandal. Although there was no evidence to support this, it caused a huge stir in the media and led people to be hesitant of vaccines to this day. So what we get from this is that the cause of autism actually isn't known at the moment, but it is likely that it's caused during development in utero. So that's prior to any infant vaccination. Another example, Daniel, I'm getting messages that you need to pay attention. The next example I want to talk about is the Alabama Tuskegee study. And this one's really serious because it was a huge violation of human rights in which the US government intentionally infected Black men with the causative agent of syphilis. So this is known as Treponema pallidum, and without their knowledge, they were infected without their knowledge or permission by the US government. This study went on for 40 years and had wide-reaching effects on the sexual partners and children of the 600 people involved. It also created a justifiable mistrust of vaccines for people of color, which is just as relevant in modern Canada. And I bring these examples up because these ethical violations contributed to public distrust of the scientific community, which I hope can be resolved with transparency and education, which is why I'm giving you guys this presentation, so that everyone may feel empowered to make informed decisions regarding their own health. Okay, I'm also checking the text to make sure everyone's behaving. Okay, thank you very much. Next slide. So I have another brainstorm. I want to ask you guys if you know the difference between bacteria and viruses. So I'll give you a chance to kind of talk about that. Plankton from SpongeBob. Plankton. That's a good point. I think plankton is bacteria. I'm pretty sure. I actually have to look that up. I don't know that off the top of my head. Let me do it. Is plankton bacteria? I actually think, oh yeah, they are. You're right. Whoever said that, it's bacteria. And they are responsible for making most of the oxygen in the ocean. And they're like the #1 thing that gets eaten. What else do we have? SpongeBob is a great reference too. Bacteria is there, and if you catch it, and viruses spread. That's an interesting one. Both of these can spread between people. Both of them cause infection, and I think I'm gonna use this as a jumping off point to get into what I wanted to say. So bacteria, Bacteria, can they grow on anything? They do have their limits. Bacteria can grow on a lot of things, but certain really extreme conditions, like a really high temperature or a really acidic environment, that would kill them. But they can survive in a lot of environments. And viruses are actually better at surviving outside of their natural host. So viruses can, well, actually, viruses have to survive within people, but they're a bit more robust. Anywho, I wanted to say that the main difference here is that bacteria are single-celled living organisms which live in a variety of environments, including the human body. Bacterial infections can be treated with antibiotics such as penicillin versus viruses, which are technically non-living microorganisms, meaning that they don't grow, they don't make their own energy, and every single part of it is made by their host that they have to infect. So they can't survive without their host or make any single part of their structure. And from here, I want to talk about the features of this SARS-CoV-2 virus that I drew here, which stands for Severe Acute Respiratory Syndrome Coronavirus 2, which is definitely a mouthful. This is responsible for COVID-19, which is the disease that the virus causes, and the pandemic that we are currently living through. And very soon, the vaccine campaign will affect all of us as, generally speaking, healthy young people in Canada. So the main features I want to point out here, first of all, we see this pink spike thing. This is known as a glycoprotein, and this you can think of as the key which fits in the lock on a human cell. So this protein attaches to a human host cell receptor, and from there it's allowed to sneak into our cells. So this is the main way that it gets into our bodies. The next thing I want to point out is the envelope, this green thing containing the whole particle. This provides protection, and you can also see these other things embedded in the membrane, and these are derived from the host cells. So once this virus is ready to explode out of a host cell that it's infecting, it will steal a little piece of the host cell membrane so that it can use it as camouflage and continue to infect other cells without being detected by the immune system. So that's actually one of its sneakiest features. It's like a cloak of invisibility. The next thing I want to show is this squiggly blue thing in the middle here. This is known as the nucleocapsid protein, and its job is to be a harder shell to protect the DNA, which is embedded within it, or sorry, the RNA. That's the genomic information of the virus. And I wanted to point out the key difference between DNA and RNA is that we have DNA, which is double-stranded, and it has the ability to proofread itself to make sure that it doesn't make a lot of mistakes, which would cause mutations and changes in our body, which could lead to things like cancer. Versus RNA is single-stranded. It's much more likely to make mistakes when it's being copied. However, its number one goal is to be copied quickly, so it being single-stranded is very helpful for the virus. And I just want to check, is this a good pace and everything? Who needs to be called out? I'll make sure they're listening. Okay. And okay. All thumbs up. You guys are listening. That's awesome. Thank you. This is heartwarming. Okay, I also have the details of what I just went over. If you want to see those, are to the left. But I think I already went through those. Oh, okay. So we're on to virus replication. I wanted to use an analogy that would make this easier to understand than all of the complicated cellular parts. So I'm going to be referring to COVID, or the virus SARS-CoV-2, as a thief that's breaking into a factory. The thief is only armed with the tools to break in and hack into the production line to change what the factory is mass producing. So the first thing the thief must do is find a way in, which I've shown here as the thief holding the keys with the lock. And once they find that lock, this is equivalent to the spike protein fitting into the ACE2 receptor on a human host cell. The next thing that the thief has to do is pick the lock, and from there, the thief can essentially walk right in the front door because they don't have to break in, they just have to sneak in through the front door with the connection of the spike protein and the ACE2 receptor. So that would be step 2. The third step is going to be the thief dumping out their bag of tools containing only the instructions to hack into the main office computer of the factory. This is equivalent to the genetic information, so the RNA. And from there, we're going to see that the computers are going to be hacked and the factory begins to mass produce copies of the tools and instructions that the thief needs to break into more factories. From here, everything that the thief needs is piled up and waiting for them right at the exit until the last step is that the thief bundles up all of their stolen goods, drenches the place in gasoline, and throws a match back so that they can escape. And from here, it's going to repeat the process, only multiplied many times within your body. So from here, it's going to spread exponentially. Speaking of spreading, my next slide is about transmission of the virus. So I drew this picture while sitting from the perspective on the back of a bus, and I'm sure you guys have all seen something like this before. I saw somebody with their mask half on just get all up in here, wipe their hand all over all the slimy parts, and then touch as much of the bus as possible. Before they got off, and I was so grossed out by that. And lately I've been imagining the environment around me more like this, and this helps me visualize the invisible spread of SARS-CoV-2 through respiratory droplets and aerosols created when an infected person coughs, sneezes, sings, shouts, or talks, according to Canada Health Services. The smaller the droplet, the longer it lingers in the air, which means the more risk you face in confined public spaces. These infectious droplets come into direct contact with the mucous membranes of another person's nose, mouth, or eyes, or they can be inhaled into your nose, mouth, airways, and lungs. The virus may also spread when a person touches another person through a handshake or a hug, or when they touch an object, such as this bar on the bus, or a pin pad at the grocery store, or an item of clothing. It could be anything. However, I do want to point out that the main difference between aerosol droplets and fomites, which is what we refer to as the virus on inanimate surfaces and objects, is going to be that 90% of infections are caused by aerosol droplets over fomites. That's because in the human body, we have way more ACE2 receptors in our lungs than we do in our digestive tract. So that spike protein can stick to our lungs a lot better than it can stick in our stomachs after we digest it. And on top of that, your stomach is really good at being an unhappy environment for anything to live in. It's just a pit of acid. So it's a lot harder for a virus to survive there. What's next? The R naught, meaning the basic reproductive number, is the measure of how many people an infected individual spreads the virus to. The Centers for Disease Control and Prevention reported that SARS-CoV-2 has a median R naught value of 5.7. So on average, an individual will pass the virus to 5 to 6 people in that incubation period within the first 4.2 days before they even have symptoms and realize they're sick. This is a really high number, and this is what makes it spread so quickly. And in some cases, we've seen super spreaders, a single individual who pass it on to as many as 100 or more people just from being very unsafe, not taking precautions, and not really caring about their impact on other people's lives. The next thing I want to show is a little video that I found on TikTok the other day while I was working on making this presentation. And I think it shows really well how a virus is turned from a virus into a vaccine and what happens inside of your body. So just enjoy.
You need to make this. What is it? It's a part of a virus. A virus? Just a part of it, not the actual virus. They're called COVID-19. They have fork hands and they're coming. Fork hands? Please. What is this? This messenger told me to make them. I've never seen these before. I'll make some new antibodies to get rid of them. Seize the forks. Nice body. Going to make this place my new home. Fork hands. Seize them. Ah! Ah!
I really like that. And also it's very scientifically accurate. So that leads me right into my next slide. And I think that there's been a lot of news that's throwing around terms like antibody therapy and vaccines. So I think it's important that you guys understand the difference between those two. So antibody therapy, which are shown in this diagram on the left, directly treats an infection and it's going to work immediately. Unfortunately, it only gives you protection for weeks to months. So the only real option to make something like SARS-CoV-2 go away for good is a massive vaccine campaign that's going to span the entire globe. So the difference between vaccines and antibodies is that a vaccine is made out of the genomic information of the virus. This gets injected into you, and then from there, your body is going to make that protein, that key that allows the virus to sneak into your cells. That then triggers your immune system to make the antibodies, which is the same thing happening in this therapy. And then from there, you have a lifelong protection from the protein. Every time that virus infects you, your body will recognize the protein and it will immediately start to destroy it, just like the fork hands guy. And I would also like to point out that the only way back to life as we once knew it is going to be through vaccination. But in the meantime, social distancing, quarantine, and contact tracing are essential steps. But overall, vaccines are the only way that we'll be able to come out of these alternating waves of going back into the public and then quarantining again. Otherwise, it'll just keep happening and people will keep getting infected. So we are looking at getting vaccinated in Canada as, generally speaking, healthy young people somewhere around June or July. Right now, we are in phase 2, which is mostly focused on treating seniors, Indigenous peoples, hospital staff, vulnerable populations, and people who work in nursing services. So we're going to have to wait a little bit, unfortunately, but in the meantime, just please try to be safe. What else? The next thing I want to talk about is mutations. So I made, obviously, this lovely diagram of the X-Men, my favorite superheroes. No cap, they are the best. And I think what I wanted to say here is that it is important to monitor changes in the virus because our current vaccine only targets that one protein, the spike protein. And if this were to mutate enough, if it were to change, then the virus would be able to sneak past our immune system. Even if you have been vaccinated, you won't be able to recognize it anymore. And as I said earlier, because its genomic information is RNA, it's much more likely to make mistakes when it's getting copied, and that causes it to be mutated and have new proteins so that it can sneak into our cells in different ways. Going back to the thief analogy, the thief doesn't care about being exact when making copies of the tools and instructions. Their number one objective is to go fast, and sometimes the sloppy work allows the thief to continue breaking into factories even after the so-called security guards are on the lookout for them. So even though we have 3 vaccines right now, Pfizer, Moderna, and AstraZeneca, that are approved by Health Canada, we still should be working towards new vaccines that recognize multiple features on the virus so that even if it were to mutate, we could still catch it. And we also need to make sure that as many people get vaccinated as possible, because even if you get vaccinated, that person who doesn't get vaccinated is putting your life on the line by making it possible for breakthrough mutations to come through the population and affect even the vaccinated population. And I think that's a really good point to mention when you're talking to people who have vaccine hesitancy, is that their decision not only affects them, but it also affects everybody else who is vaccinated. And on a more personal note, this started as a way to teach you guys a little bit about immunology, which I think is really cool, and I hope you guys are more curious about after this. But it became a lot more serious to me when 3 out of 4 of my housemates were diagnosed, that sounds so serious. They were tested for COVID and confirmed positive. And since then, it's just been really stressful in our household. We've had to go into complete quarantine, and all of us are in self-isolation in our rooms right now. We don't really know how it snuck into our house, but we do know that we were getting way too comfortable expanding our bubble and going out to restaurants. And it could have been from something just like touching the wrong pin pad somewhere, but I do know that the coughing, the sneezing, the fatigue, the headaches, the stress that we're all living through right now is not worth it. And I don't want any of you guys to have to go through that. And I do know that it is directly linked to the risky behavior that we were partaking in. And I don't want to blame anyone for getting sick, but you can also take a more active role in keeping yourself healthy. And that became a lot more serious to me in the past few weeks. So I hope that you all wear a mask, wash your hands frequently, keep your bubble small, and please get vaccinated. And that's my presentation. I want to take your guys' questions, and I also have a Kahoot. Dun, 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 dun. Woo! I also realized I look like Miss Frizzle, so I just really embraced it with the lab coat. I just wanted you guys to know. Let's see. Stop share. Oh no, I need to keep sharing.